Thank you. Fantastic introduction. And that day, about this time last year, was a great learning experience for my group, which includes some junior doctors, but mainly PhD students, and to meet the people that we study and all, talking about in an abstract sort of way. That was a fantastic, memorable experience for them. They worked very hard. They got a series of blood reports back to that group of patients. I think we collected blood from 20 or more patients, with informed consent provided, of course. They went through all the ethics. And then poor Phil Choi, who was our lead hematologist on that day, had to interpret our biologically facing reports on platelet function to the people. Basically, what does it mean? So that's a challenge in itself. But I'm really pleased to be invited here today by Danielle and the crew. And so I'm not a hematologist. I'm a platelet biologist. I try to understand molecular processes by which platelets come together and form a blood clot. So Danielle's asked me to just talk a little bit about, platelet function, why it's important. Well, I kind of feel a bit shy of this title because I think I'm talking to a room full of people that absolutely understand how important platelet function is. This is a slide actually I give to third-year students where I talk on thrombosis hemostasis, and so always sort of start off with this slide because it shows some beautiful images of platelets under resting or activated conditions. So in that upper left-hand panel, it's actually quite hard to get a photograph of a resting platelet because as soon as we take them out of the bloodstream, they're next to a plastic or they've gone through a steel needle. It's very hard to capture a platelet in its resting state because it's already amped up. And on the right, it's quite easy to get photos like that. So they are scanning EM images of a resting and an activated platelet, and we can trick a platelet into becoming more activated in the lab. So we do a lot of tests taking healthy donor blood and making it activated, and then we do many different sorts of readouts to understand those molecular pathways and try to modulate them, try to control them, because if you have too much clotting, that is also a problem, as everyone here knows. But, for the purposes of today, we're trying to see how can we trick a platelet that is maybe less functional, and that is a key project in our group to understand in ITP what's the platelet functional capacity in people with low platelet counts. And it sort of traces back to that, I know it's Danielle's mantra that it's not just about the count, the number, it's also about the function. So, two people with a platelet count of 50, one may have real bleeding issues, anything from blood nose to having a tooth pulled and it bleeds and bleeds for days. And that other person also with a platelet count of 50, going through the same sort of traumas, doesn't bleed. So it's trying to understand what that, try to understand on a molecular level and help doctors make some decisions around therapies for those two people that have exactly the same platelet count but are both at risk of bleeding. Inside the platelet, it's a little cartoon image from those scanning EM photographs in the lower, just to sort of emphasize, platelets are not just little socks of membrane. They are full of granules. There's at least 3 different types of granule. They have their own glycogen store. They have mitochondria, which are like the powerhouse of the cell. That's where the energy is made for the platelet that the platelet draws on for its function. The only thing they don't have is genomic DNA. So they have everything else, and they can make new protein. They just don't have the genetic material because they don't have a nucleus, which presents some challenge to studying them, but it also makes them quite convenient to use and handle in the lab because if we ever want to break them open, if it was a full cell with DNA inside, it turns into something looking like chewing gum very quickly because of that, all that DNA that's in a cell that has a nucleus, whereas platelets, we can handle them quite readily. So this is a little movie just to show where platelets are made. And so I'll just let you watch this. The colour code is on the bottom here. You may not be able to see it, but I've put it on the next slide. And this is a snapshot of a piece of bone and showing where the platelets are made. So the parental megakaryocyte that makes platelets sits in all of our bone, and we sort of think of bones in the legs and the hips, but actually the sternum, in the cranium. Megakaryocytes live close to the sinusoid, so close to the blood supply to the bone. And so what the point of this little video, this is not from some colleagues of ours that have published it in Nature, is to look for the immature green megakaryocytes versus the yellow mature megakaryocytes, because it's the yellow ones that make the platelets, and just try to understand where they sit in the marrow. So on the next slide, I just took a snapshot so that we can, from that previous image, and that explains it a little better. So there's the colour code there. To orientate you, blue is bone. The grey are the veins, or the blood supply to the bone. So wherever you see grey, that's filling in where a vein is. Blue is where the bone is. And then the yellow and green colours represent immature, sort of young megakaryocytes versus the yellow mature ones. And so what this paper was talking about was how close to the sinusoids the yellow colour is. And those, and that you can see they even wrap around the sinusoids. I don't know if I've got a, it's like there's a mature megakaryocyte sitting right on a sinusoid, whereas these green ones are sort of more, not free floating, but more distant. And we can measure this, we can make measurements to determine how close is a yellow to a blood vessel versus a green. And this is important, and in this paper they went on to sort of begin to treat treat the system so that to generate more yellow megakaryocytes to try and amp up platelet production. That was kind of the point of the paper. But I just thought this is a nice image just to show where the platelets began to be made by the mature megakaryocytes, and they extend tentacles into those sinusoids, and the platelets bleb off, if you like. And so another major important facet of platelet function is to understand where the bleeding actually occurs. So this is another colleague who's published it recently in a scientific journal. And he was, there's a theory, it's more than a theory now, out in the world that I work in, around platelets are not all they're not all equally functional, and they're not all with the same designated task. So they all have this task around hemostasis, so to minimise bleeding, to limit the loss of blood, and they do that by forming aggregates. But this is just a nice image, so I'll take you through several images, but again, they're colour-coded, and in this group are talking about sparse platelets versus densely packed platelets, and I can explain a little bit how a platelet would go from being sparse to dense based on its the receptors on the surface. And in these images, the red blood cells are coloured red, the white blood cells are coloured in blue, and if we just look in here, this is intraluminal, so this is within the blood vessel, and this is extraluminal, so this is where an injury is being created in the vessel wall to generate a bleeding response. And then using specialised electron microscopy, they were able to capture what the thrombus looks like and colorise it. So long and the short, there's two types of platelets, and in this particular injury, you can see that there are clear regions where the Red cells are encapsulated around densely packed platelets, but out here in this region, a little more away from where the site of injury is, we have these sparse platelets. And this has important implications for how we might treat someone that is having a thrombosis, depending upon where that thrombus is forming. But it also is an opportunity, in someone where there is either a low number of platelets or the platelets aren't working particularly well. Can we treat either the blood vessel to enhance the ability of the platelets to form an aggregate and prevent blood clotting, or in some way treat the platelets themselves to amp up their ability to respond to an injury? So this is all very detailed, and all I wanted to show you was these are the ways that we can study platelet function. And so to just bring home that message around platelets behave differently, whether the injury is in a venous system, or an artery system. And so I just picked a couple of images out of the paper. And from hundreds and hundreds of these sorts of images, we can show that depending upon where the injury occurs, either in the venous system or an arterial system, the platelets behave differently. So in one system, there seems to be more red blood cells incorporated, in the venous system, but in the arterial system, almost no white cells are detected, but an enormous number of the densely packed platelets versus sparse. So this, I don't want to belabor the point too much, but platelets behave differently depending on the site of injury, and this has implications for how we would treat people who are at risk of clotting or at risk of bleeding. So this is just one of my old favourite slides. I also give this in lectures too. I've worked on these two major functional receptors on the platelet surface, and we essentially try to understand the signalling pathways that turn that resting platelet into a platelet that can now be part of a blood clot. And we do that with all different sorts of techniques. This one's a good old-fashioned Western blot. I love showing Western blots. So we can look at, this is the receptor for collagen on the platelet surface, and we can show that if we treat the platelets in a certain way, we can get the platelet to release this receptor. So now the platelet becomes less responsive to collagen. So I won't sort of belabor this point, but we do a lot of these sorts of analyses, and these are all human platelet studies. And there's a group of people with ITP where the antibody actually targets this receptor. So they have anti-GP6 antibodies. We don't know why, we don't understand what happens, but the way the hematologist picks them up is that the person no longer, their blood sample no longer responds to collagen in an aggregometer. And that's because this receptor has gone missing because the antibody has blanketed it. So now the platelets just can't respond to collagen. So there is a strong ITP point here. This was just really quick. The other system that I use where we have, just to orientate, we have a confocal microscope and a video camera all hooked up to a computer system. We have a stage, and on it is a microscope slide that we've covered with collagen to begin to look at that collagen receptor on platelets. We have some blood sitting in a little reservoir here, and there's a pump that actually pulls the blood across the collagen surface, and we can set this rate however we want. And then there's a little reservoir of buffer just to mix the blood in. And so we can study how a blood clot forms. This is actually looking down the microscope onto that slide that's covered with collagen, and these little guys here, these are platelets. And so what they're doing is they're immediately sticking to the collagen. This is not sped up at all. This is a real-time event, just showing as the blood is drawn across that collagen surface, the blood, there's enormous numbers of red cells and white cells here, but it's only the platelets that are sticking to the collagen. They're adhering to the collagen. We have to use something called Hiridin, which is to block thrombin, because if we allowed thrombin to be generated here, we wouldn't be able to see anything at all. We'd just get an enormous clot straight away. But the points to make are, it's fast, it's specific for platelets, and it's very controlled too. In a normal platelet function is very controlled. It's not like the channel fills up with thrombus straight away. So that's, and that work from our group has shown how critically important some of the platelet receptors are for this process. And, we're looking at ways to modulate this, either to increase it in someone at risk of bleeding or decrease it in someone with a thrombotic risk. So the take home is simply that the platelets come from megakaryocytes. Platelets are released into the bloodstream where they circulate in a quiescent form unless they meet an injury. If that's normal hemostasis, but if it doesn't occur appropriately, we can get either bleeding or clotting phenotypes. And that's essentially what I work on. So using this system, we can do all sorts of really nicely refined manipulations in the lab. We can take sections through the thrombus as it forms, and we can take photographs of what's happening in different levels. We can stain them for different stains, for different antibodies to pick out where glycoprotein 6, the collagen receptor, is, for example, and where is it strongly found and where is it not so well found. So this is one example where there's 3 different colours in this on this slide, and that's a platelet dye, so we just labelled all the platelets green. And then we looked for 2 different receptors, glycoprotein 6, the collagen receptor. This is another receptor, and using the antibodies tagged with the red colour, the one that finds this receptor, or it's tagged with a blue colour if it's looking for this receptor. And then we can pile them all up and we get a beautiful rainbow plot. But if now I take away the green, I take the green out of the image, we can see that there's different areas where the pink is, or the red is, versus the blue. And we can see the outer portion of the thrombus is really rich for the collagen receptor, whereas the inner portion less so. And that's probably due to the way the receptor is controlled on the membrane of the platelet as it goes into a prothrombotic response. My point is essentially to say we work on a lot of these sorts of problems, trying to understand what happens normally and then what happens in people who are at risk of clotting or bleeding. So this is just the last little point, a paper from our own Phil Choi. He's led this study looking at ITP patients. The overwhelming majority of these patients are his. And this is just looking at some platelet parameters and the way the blood behaves. And you can just see, you don't need to know any of this detail, just wherever you see red, that is a difference between the yellow ITP group and a control group who were people with low platelet counts but not due to ITP. Some of them had had chemotherapy, or they had genetic-based thrombocytopenia. But the take-home is we now have that ability to tease out, and we can see there's big spread. So ITP patients, these are all people with primary ITP. Some of them are behaving like the blue group, the healthy donors, and some of them are not, and depending upon which test we look at. So just to let you know, these are the sorts of studies that we're now deploying, and we were able to take some of this work, and again, getting back to that question about people with a low platelet count, who's going to bleed and who's not. So now using this simple test, if we just focus on this part on the right, these are all people with low platelet counts, going from down to nearly 0 to 50. So they've all got thrombocytopenia, but the ones that are symptomatic are coloured in black. So these are people who are bleeding or have had a bleeding event versus the asymptomatic. And so you can see here, we can separate out now and we can identify using some of these new tests and begin to evaluate the people with the low platelet count who are bleeding, for example, at a platelet count of 20 versus someone here also with a platelet count of 20 who is not bleeding. So that's sort of the thrust of the laboratory work, and I just wanted to leave that with you. And this is work, I think we're going to hear from Phil Choi. This is work that he's leading with our group in Canberra. So it just remains to thank the people involved. This is just my group who did all the work. I just organised the slides. Lots of students, former students and postdocs, and of course it would not be possible without Phil Choi and other members of the ANU group. So I'll leave it at that point. Happy to take questions if that was something people wanted.
It's Mark. I'm a patient. That last point you made, that test, is that a diagnostic test you can do at the moment, or it's just a laboratory-based test? And is it useful for people like me who don't bleed but have got a low count?
Yeah, so the test is diagnostic. It's called a ROTEM, or a viscoelastography. It's not ratified for people with a platelet count below 100, which is, pretty much every ITP patient. So it's not, it's, useful to look at platelet function, or, the surgeons often use it in surgery to decide whether they've got a deficiency, whether the patient needs to have some frozen fibrinogen, fresh frozen plasma, or a fibrinogen concentrate. They use it as a tool to assess bleeding, but it's not used in people with low platelet count because it hasn't been standardised. But in the research setting, we can do that, and have all the caveats in place, and the doctor does not make a decision based on our findings. But in the future, I hope we will be able to standardise it for people with lower platelet counts, and it can be used to help fish out. So, and it's just one of a battery of tests that we can perform in the lab that are not yet diagnostically available. Yeah.
So the narrative from my specialist is, well, Mark, you've got low platelet, and it's sort of 30 to 50, but they're really big and they're young because they're growing quite quickly because obviously they're turning over.
Yeah.
So and I can't remember the term, but they're like way outside the boundary in terms of big. So they're really big. I'm just interested in any research or your thoughts on that, because would the bigger ones tend to be more sparse, or how does all that work?
That's we can't answer. But so immature platelets, or platelets when they're born, they have a certain size, and as they circulate for 7 to 10 days in healthy humans, and they begin to lose their membrane, they bleb things off, and so they do shrink a little bit. And so the hematologist will get a mean platelet volume in a blood sample, and which is considering the volume of of millions of platelets. But in someone with a low platelet count, it is known that there'll be, because the bone marrow is under pressure to produce platelets, it pushes the platelets out probably in an immature form. So I would argue those platelets also probably are not replete with all the receptors necessary to perform the function. So the person not only has a low platelet count, they also have platelets that are not quite replete. So these receptors all work on being at an appropriate density. And so if you've got a larger platelet for the same number of receptors, you can imagine that they'll be spread out and not quite as functionally strong as someone with a complete repertoire of young platelets and old platelets circulating through the system. The platelets that were just born, and we make about 1 billion platelets a day. Versus those that were born yesterday or 2 or 3 days ago that are still circulating, unless they're involved in, a hemostatic response. So, it's okay. Thank you.